Hey guys, welcome back to the channel. In this video, I'm going to be taking you through a full course on how to build an R-Shiny app that displays data from a single cell analysis with the R package Seurat. The thing is with R-Shiny is that there's so many applications that this can be useful for. Like today, we're just building a single cell analyzer app where we can look at our single cell data, which by the way, is very useful. Like from my own personal experience, I found that having a way to quickly pull up data and make connections is invaluable. It speeds up the rate at which you do research massively. I'm going to start by teaching you the fundamentals of R-Shiny. We're then going to go into some single cell RNA sequencing analysis before finally we actually build out the bulk of the app. This video is meant for anyone who's interested in building your own R-Shiny app. It's a great project to put on your resume if you're trying to display your skills. Now I'm hoping you have at least some R experience because we are obviously building an R-Shiny app, so there is that prerequisite. But aside from that, I'm assuming you have absolutely no knowledge of R-Shiny. I'm going to be teaching you the fundamentals and we're going to progressively go more in depth into actually how to build out the different components of our app. So if you're interested, then buckle up because this is going to be quite a long video. But without further ado, let's get into the video. Okay, hey guys, before we start, I just want to let you know all the code for the dashboard that we're going to be building today is going to be available on my GitHub. So I'm going to link that in the description of the video. I encourage you to actually follow along and code alongside me because I think you're going to better retain the information and better kind of learn the concepts. However, if you just literally want to, clone this repo and get the app up and running straight away, feel free just to go to my GitHub and clone it. It's private at the minute, but I'll set it to public before I post this video. Anyway, so let's get into it. Firstly, I want to show you what this app actually looks like. So this is what we're going to be building today. It's a very simple R-Shiny dashboard that's going to allow us to view and analyze some single cell RNA sequencing data, specifically data that's been generated from an R package called Seurat. So Seurat is a very well-known package for doing single cell RNA sequencing analysis. And so basically I'm going to take you through some of the basics of single cell RNA sequencing. But the main point of this video is to show you how to build the dashboard. So we're going to be displaying some of the results, some of the plots generated from Seurat in this app. So let me give you a quick demo. So when you open the app, you're going to come to this welcome page. Welcome to the single cell RNA-seq Seurat analysis R-Shiny app. Now this is very plain, but we can go to the sidebar on the left here. We click on single cell RNA-seq analyzer, and once we click on this tab, we will initially see these instructions pop up in the main panel, so telling us how to use the app, and we'll also see some extra inputs in the sidebar as well. So if we just look at these instructions, upload a Seurat object using upload file. So that is basically here. We click browse. We can upload a Seurat object file from our file system. I've got one right here conveniently. So we can upload that, and it's quite a large file, so it will take maybe 5 seconds to upload. But let's just go through the rest of the instructions. So we can press run to run the analysis. 2 tabs will appear with the data we can explore, and then we can press the reset button to clear the input files and remove all tabs. Okay, so once that's uploaded, we can click run, and we'll have this pop-up widget while it's loading. And there we go, so it's loaded, and we basically have 2 extra tabs that have appeared, one called UMAP and one called gene expression. So we can easily flick through these tabs, and what these are basically showing, these are both showing UMAPs. So if you're familiar with single cell RNA sequencing, you'll probably know what a UMAP is. It's basically a visualization of our data in a dimensionally reduced space. So we're showing the variation of our data where each dot in these graphs is a single cell. And we're showing, I guess, two axes of the most variation in our data. If you don't really understand that, it's okay. We're going to go a bit more into single cell a bit later on, but let me just give you a demonstration of what we can do. So in this UMAP tab, we can basically display different metadata of our Sora object over the UMAP. So here we look at the number of counts, the number of features. We could also look at the cell types, for example. Whatever metadata columns we have in our Sora object, they're going to appear here, and we can easily display them over our UMAP. We've also got this download UMAP button beneath the graph, so if you click that, it's literally going to download the UMAP for us, and it's going to name the UMAP after the metadata column that we're looking at. So for example, here we're looking at sex. It's going to name this sexumap.png. So yeah, as I mentioned, each one of these dots is a cell, and we can see each cell is coming from either a male or a female in this example. Let's see, so we've also got this gene expression tab. So what this is showing is the expression of a certain gene. So any genes that are in our Sora objects, they're all here. We can see the expression of any gene over in the cells. So these are all quite lowly expressed. We can just pick a random one. Let's see if we can get a better one. I don't know, but we can also type here. There's so many genes in our Sora objects. It's going to take ages to scroll through and find it. So we've also got the ability to search for genes as well as type them in as well as search for them. And if there's no gene that exists, it will come up with this gene doesn't exist over the top. And then a similar thing, we can download this feature plot, and it will be named after the gene again. So it'll be gene feature plot, whatever your gene's called. And then the only other feature I want to show you is this reset button. So when we click this, the tabs will disappear, and also the file that we uploaded will disappear as well. So this is a very simple RShiny dashboard, but it's actually very useful. So I used to work in a lab which specialized in single cell RNA sequencing. And a lot of the time when you're doing research, you want quick answers like, is this gene being expressed? Like, what's the distribution of, whatever metadata you want to look at, or how are the cell types being displayed? And oftentimes, if you're producing these UMAP plots, it's a very manual process. You have to go in, edit the code, type in, for example, with the feature plots, type in what genes you want to display, and then save the plot, and then open the plot and look at it. Whereas something like this, it's going to allow you to really quickly look at your data. So it basically is speeding things up. It's allowing more efficiency, and this is something which would have helped me at the time when I was working in that lab, to increase my efficiency in finding results. So this is what we're going to be building today. And the structure of this course, we're basically going to start with the fundamentals of RShiny. So I'm going to be assuming you've not built an RShiny app before. I'm hoping you are going to have some experience with R, because a lot of the programming we're going to be doing today is in R. But that's about it in terms of prior experience. So we're going to start with the fundamentals of RShiny. Then we're going to go through a very basic single cell RNA sequencing tutorial in Seurat. Excuse me. So actually, we're going to follow the tutorial on the Seurat website. It's a very basic run through of how to produce the Seurat object that we're going to be using to display the figures on this app. Now the tutorial goes into a lot more detail on their website, so I'm kind of going to breeze over it because I want this course to mainly focus on building the R Shiny dashboard rather than the single cell. But I will share all the links, so if you do want to go into more detail on the single cell stuff, be my guest, go ahead. So once we've gone through the single cell, then we're going to come back to R Shiny and we're going to build out the bulk of the app. So without further ado, let's get into it. So if you've ever thought about building an app before, you probably have the same thought that I did, is that that's a really complex thing to build. Like, you've got to learn front-end frameworks, back-end frameworks. You've got to have, a server, a database, maybe an API connecting the front end to a back end. There's so many elements involved, and it seems like this massively complex thing. However, with R Shiny, we can effectively build a whole app, front end and back end, just using R. So just using R code. So it's quite brilliant in how simple it is. And this right here is the structure of every R Shiny app. There's three main components. There's the UI, the server, and then this function Shiny app which calls the UI and the server. So the UI is our front end. This is where in our dashboard we're going to build out our sidebar and our main panel and whatever features you want. The server is the back end. So this is going to be the actual code that's going to be running behind the scenes. So when we are going to produce a plot, for example, and we want to plot a certain gene over our UMAP, all that kind of back-end programming, that's going to be going on in this server block of our code. So this is a very basic overview, but this is pretty much fundamentally how every single R Shiny app is structured. Now, there are different types of R Shiny apps you can build. In this course, we are focusing on building an R Shiny dashboard. So a dashboard is a specific type of R Shiny app that has its own structure. And that's what this is showing. So here we see the UI. In the UI section here, we have basically a dashboard page. And within this dashboard page, we have three elements: a dashboard header, a dashboard sidebar, and a dashboard body. So if we're building an R Shiny app, well, at least with the Shiny Dashboards package, which we are, this is going to be the structure. It has to have this dashboard page and then three components inside it. And then the server is independent of the type of R Shiny app you're building. So a server always has an input and an output parameter, and then it has this third optional session parameter. So the input is basically everything we as the user of the app are inputting as inputs into the app. So what I mean by this is, let's say we are, for example, in the demonstration I showed you, we're uploading a file to our R Shiny app. That is an input. Or we could be maybe moving a slider from 0 to 10 and we're setting a value. Maybe we're inputting a number, or we're selecting an option from a drop-down list. All of these are inputs into our app, and this is what this parameter represents. Then we have this output parameter. So this is kind of the opposite. So this is the output that is displayed on our app. So we have kind of static components, but output is more to do with things that react to the input. So let's say our input is, let's say a certain gene that we want to see on our UMAP. That's our input, and then the output would be the plot, which is displayed on our app, showing the gene expression. So anything that's displayed on our app as a result of the inputs. And as I said, session is an optional parameter. It basically sets like the environment. So we didn't even need to include this in the server function because we're just going to be developing it locally. So that doesn't matter. I wouldn't worry about that. And then again, we have this Shiny app function at the bottom. This is basically how we run our app. So it's calling the UI in the server, combining them together. And once we run this, we will be able to view our app on. Actually, yeah, let's cover this because this is probably something which is not that obvious. Once we run our Shiny app, it will basically provide us with a local web address. And let me just quickly show you what that looks like. So here, so listening on local IP address, this is your local host. This is like a common thing in like web development. If we just copy and paste this into our browser, it's basically hosting it on our own machine. So if I just copy and paste this into our browser, it's just going to come up with the app that I just showed you. Right. So we've covered the basics of our Shiny. There's three main components: the UI, the server, and a function which calls the UI in the server. Okay, hey guys, I'm actually recording this part of the video from the future, or should I say later than the rest of the video is recorded. So I kind of breezed over the single cell tutorial to begin with, but I thought it'd probably be worth going back and going into it like a bit slower and into a bit more detail, just so that for those of you who haven't done single cell, which you know a lot of you might not have, because this is an R Shiny tutorial more than it is a single cell tutorial. But I figured why not just kind of do both, try and teach you this properly. So yeah, the reason we're going to do this is because we need to generate the data that's going to be shown in our app. So we're going to be displaying some single cell sequencing data. Specifically, we're going to be showing some UMaps. So a UMap it's a form of dimensionality reduction, and how I can explain it is like within our sort of count matrix. So we have a count matrix where we've got like rows and columns are the genes and the cells, and so in each cell you have an expression value of a gene, and you might have thousands of genes. So each cell has an expression value for all these different genes, and we want to look at how our cells differ based on this gene expression. And that's obviously very hard to visualize because we have, let's say we have 20,000 cells and 20,000 genes. There's so many like points of variation to look at. Like it's a hard thing to visualize. And a UMap it's a way that we can look at the variation within our cells in 2 dimensions. So I'm just going to search in Google UMap, see what comes up. This is just off the cuff. And I'm hoping, yeah, so this is kind of what it looks like. It's basically loads of dots on a graph, where each dot represents a cell. And generally, cells that are similar are going to cluster together because cells that are similar have similar gene expression. That's kind of the high level overview. I'm not going to go into this too much in terms of like how the UMap works, because that's more like mathematics. That's quite complicated, like statistics. But basically, it's going from thousands and thousands of dimensions to just 2 dimensions. So we're looking at a 2D figure here. So we're showing variation in 2 dimensions. That's basically all a UMap is, and this is what we're going to be generating to show in our app. So we're just going to be going through the Seurat guided clustering tutorial. So this is on the Seurat website. Seurat is the main package in R for doing single cell analysis. We're going to be going through this tutorial just to keep it simple. So the first thing we're going to do is actually download the data. So if you're on the same page, and I'm going to share this link in the description, but you'll see like a hyperlink here. The raw data can be found here. I'm going to click that and download that. It's about 300 megabytes, that file. So this should just be fine to do on your local machine. So just on your personal laptop. That's what I'm doing. So you want to download that, and you'll see it's downloaded. And once it's downloaded, I want you to create a new folder in your, just on your laptop. Create a new folder where you want to build this app. So for me, I've just created a folder in my documents, and that's where I'm gonna build this app. So once you've downloaded the data, basically just move it to the folder which you've created where you want to build this app. So I've just created this folder. I've copied the file that I've just downloaded from the website, and I've just pasted it in here. So about 7,500 kilobytes. And that's the zipped file. So we actually need to unzip this. So if you click on it, right click on it, and do, I use like 7-zip, just anything, like any way you can extract a file. So I'm just going to do like extract files. I'm just going to click OK. So that's created this folder here. I think I need to do it again. I need to click on it. Extract files. Okay, so now we've got this folder, and we should find 3 files. Yeah, barcodes, genes, and matrix. So these 3 files are the precursors for creating our count matrix. And actually what I'm going to do is, we don't need it to be in these many subfolders. So what I'm actually going to do is, I'm going to take this filtered genes, BC matrices, and I'm going to go up 1 folder to hit that to here, to the actual single cell RShiny app. I'm going to paste it in here. I'm actually going to delete these 2, just because all we need is these kind of these files. So there's no point having like a massive, like a folder within a folder within a folder. We may as well just have it like a shorter path. So once you've done that, let's go back to our tutorial. We're basically just going to go through this together. So the first part, set up the Sura object. So the exact data we've downloaded, it's from peripheral blood mononuclear cells, PBMC, freely available from Text Genomics. So we've got 2,700 single cells sequenced on the Illumina NextSeq 500. So this is a small Sura object, which is good. We don't want to be handling such large files, especially when we're building this app, because there's going to be like large file upload times. We want to keep that to a minimum, especially when we're just developing the app. I would even go as far as to say it would be good to create a test data set that's even smaller than this, but we're not going to worry about that today. Okay, so I'm not going to read this word for word, but basically we're going to first load in those 3 files and create a count matrix. So I'm actually going to copy a lot of this code onto my own personal R file, which I've created. So I'd say, yeah, open. This is a text editor. If you're not familiar with like a text editor, it's just like a place you can do coding. This is Virtual Studio Code. I'm just realizing, like, you probably know that if you're doing this tutorial, because okay, whatever. Open your text editor of choice. In fact, open your text editor in the same folder where you put this downloaded dataset. So for me, it's just here. So create an R file in this folder, and this is where we're going to build out the app. Okay, so let's start. First thing we're going to do is load in some packages. You may not have these packages installed in your R environment. Okay, so the first thing we're going to do is we need to open R. So what I'm going to do is I'm going to just try and open it in VS Code. I know this is quite crowded on the screen, but I feel like we need to have it all on the screen. Okay, so what I've done is I've just opened a terminal and I've gone into the R console. here, and then up here we're just basically going to be copy and pasting from the tutorial, but we're going to be changing it slightly because we have to set the right paths and whatnot. So yeah, let's go. So yeah, I'm actually going to zoom out a little bit. So this is the first step here. I'm actually going to make it full screen for now, just because it's easier to see. And the first step you see is, loading in the data set. So we need to change this bit to the path where we downloaded it. So what I would do is basically go to the H19 folder, right click, properties, and then the file location, you want to copy that. In fact, that doesn't go all the way, so go to security and then copy the object name like that, and then we can paste this into here, so like so. One thing to note is that for Windows you have backslashes, and this will cause an error, so you basically just want to double up these backslashes in Windows. That's one way to solve the problem. I think the simplest way, so let's just do that. And I think that's fine. So what we're going to do is basically just going to copy and paste this into our R console. So I'm going to load in these packages. Okay, so they've loaded in for me. You may get an error if you try loading these packages, which could just mean you don't have them installed. So to install a package in R, you probably know how to do this if you're watching this tutorial, but you just use this command here, install packages. So and you use quotations and any package which you need to install. So for example, if you need to install dplyr, you do install.packages and then in quotes dplyr. So if you do get those errors, that's how to solve it. Right, but we're going to carry on, and we're going to try and read in. We're going to try and create our count matrix. So read10x is a function built into Seurat, and we're basically pointing it to a folder that contains 3 files: genes, matrix, and barcodes. So let's see if this loads. It looks like it's loaded fine. I'm actually going to move this into the editor area, have it side by side. Okay, so yeah, and actually what I'm doing, I'm going to print it out, and we should see it's actually like a matrix, like a sparse matrix. So I'm going to print it out. Yeah, and the sparse matrix means that there's lots of zeros. Like when you do these experiments and you're looking for gene expressions within each cell, probably like most of the genes are just going to be expression is 0. So this is what we call a sparse matrix. So that's going to be printing out a while. I'm just going to stop it by pressing Ctrl C. And the next step is to create a Seurat object. Okay, so again, this is a function built into the R package Seurat, and it takes our counts, which we've just created as a parameter, and it just sets some other, other parameters. So let's run this. And it says, warning, feature names cannot have underscores replacing with dashes. So in a Seurat object, a feature is a gene. It's the same thing. So as you'll see, like some of these names have dashes in, but I guess some of them would have had underscores in, and they would have been replaced with dashes. So if we print out PBMC, it doesn't print the whole matrix this time. It just says, look, it's an object of class Seurat, 13,714 features, genes, across 2,700 samples, cells, and 1 assay. And it's RNA. This is RNA-seq. You can have other. I'm pretty sure you can have other data types in there as well. But the thing about Seurat objects is, as well as having the matrix, it also has metadata. So you can look at that by doing the object. So in our case, PBMC. Then you do an @ symbol, then meta.data, and this will print out something large. Yeah, so I'm just going to stop it. But if we print out the head of that, which is just like the header, we see that it's basically got information about. So yeah, so this is where we can store information like about each cell. So this is a cell barcode, or the cell name, and then for this cell, this is like the data set it comes from, the number of counts, the number of features. Okay, so let's get back to the tutorial. So yeah, it's basically just walking us through exactly what we just did. And yeah, I encourage you guys, if you are not familiar with single cell, you can just follow the tutorial yourself and read through it thoroughly. I'm going to skip over some of this and kind of explain it in my own words just to save some time, but the next step is to find the number of mitochondrial genes. So why is this important? I'm just going to copy and paste it. So when we're doing the actual sequencing itself, if there's a high percentage of mitochondrial genes in the cell, that means the cell might be dying, because it means the cell might be going through like metabolic stress. Mitochondrial genes start with MT. So what this is doing, so percentage feature set is a function built into Seurat, and we're just seeing what percentage of our genes are coming from mitochondrial genes. So I'm just going to run this command, then I can show you. So if I run head metadata again, we now have a new metadata column where for each cell, so remember each row is a cell, for each cell we now have the percentage of mitochondrial genes in that cell. And we can see 3%, 3.8%, 0.8%. These are all quite low. This is good. I'd say it kind of depends like what data set you're using, but excuse me, anything under sort of 20% or 25% is probably like a good amount to like keep. You can visualize these, so you can visualize the number of counts. This is like over your whole data set, the number of features and the percentage of mitochondrial genes. You can visualize them. We're not going to do that today, but again, like these are things that you could show in your app if you wanted to like extend the functionality, but these aren't that important. These are kind of just like QC, like quality control, which you do before generating the data set. But the next thing we're going to do is normalize our data. So we're going to copy this one, and we'll also copy the one below it. Okay, sorry, no. We're just going to bother with the second one. We don't need to bother like saying exactly how. Yeah, so we're going to normalize our data. If we do this, it's a perform log normalization. It's a very quick process. And then we'll go into our next step, which is identifying highly variable features. So what is this? So as I mentioned, in your Seurat object, in your data set, you might have many cells and many genes. Let me just remind myself, how many genes do we have? We had, yeah, so in our Seurat object we have over 13,000 genes. And to be able to look at the differences between the cells, we want to look at how the cells vary in terms of gene expression. So a certain cell type's expression more of one gene compared to another cell type. We want to kind of look at that. But as I mentioned, most cells, they express genes at 0, so they're not expressing them at all. So we only really want to compare genes that are highly variable, so that lots of different cell types are expressing them at different levels. That way we can compare them on like a expression kind of level. Hope that makes sense. That was a little bit mumbo jumbo, but basically, yeah, we want to find the most variable genes in our data set. So I'm just going to copy this whole code block. Yeah, and what you can see is selection method, that's kind of algorithmically, but the one thing you can pay attention to is like n features equals 2000. So in this command, we basically want to find the 2000 most variable genes in our data set. So we'll run this. And actually, before I click enter, you'll just notice zero variable features. So this is previously before running this command. But if I run the command, it says calculating gene variances. So now if we look at our data set again, now it says 2000 variable features. So that's a good sanity check to make sure you've run it. And then we can just look at the top 10. So if we print that out, these are our top 10 most variable ones. And then the plotting, we're actually not going to do the plotting, so I'm going to get rid of that. So we'll go back to the tutorial, but this is what those plots look like. So we've got an average expression along the bottom, and then standardized variance along the top. And we can see the ones that vary the most are going to be like higher up in the y-axis, and again the expression on the x-axis. So as you can see, those 10 we printed out, I believe those are these 10 that are labeled here. So next thing we want to do is scale the data. So this is basically shifting the expression of each gene. So the mean is 0, the variance is 1. So there's a lot of like kind of standardized like normalization and scaling steps that are kind of well known and like standard procedure when doing this kind of analysis. This tutorial goes into detail about why we do everything, but I'm just gonna pretty much just copy and paste a lot of this just so we can generate the actual data. So again, scale data is the command in Seurat. Build does like scaling for you. So let's just copy and paste this. So as it says, centering the scaling matrix. There we go. And in fact, we didn't even need to do that. We could have just done this simple step, which is even simpler. I'm just kind of breezing through, so I didn't even realize, but that's just like, does the same thing, does exactly the same thing. Next, perform linear dimensional reduction. So PCA, principal component analysis. So what this is kind of similar to UMAP in the sense that we're kind of reducing the dimensions in our data. So as I mentioned, it's basically the same thing. Like, we're going from a very high dimensional data set, loads of genes, loads of cells, and we basically want to look at our data in 2 dimensions, because as humans we can only really process 2-3 dimensions. So I'm just going to skip down a bit, but this is a PCA plot, so very similar to a UMAP plot in how it looks. So we have two axes of variation, and then each dot is a cell. So I'm not going to go too much into this, but I'm just going to copy the run PCA, and it doesn't run PCA on all the genes, it only does it on the variable genes. So the 2000 variable genes we found. I'm just going to copy and paste this. This might take a minute to run, but once this is run, you'll see kind of output that looks a little bit like this. So we see principal components, and this is like a load of genes which are kind of responsible for the variation in that component. Let's not go too much into that. Let's go back to the tutorial, and now we're pretty much going to skip past all this, because this is kind of just exploring the data, looking at genes, variable genes, whatever. We're going to skip all this. We're even going to go straight past this, and we're going to go on to clustering. So we basically will now cluster our cells. What I mean by that is we want to group cells that are similar together and give them a label saying these cells are similar. We could give them a label of the actual cell type, but we'll get onto that later. So we're going to cluster cells. Feel free to read through all this, but I'm going to skip straight down to here. So our next two commands, find neighbors and find clusters. So this is again built into Seurat. This is going to do some like nearest neighbor, so some basic kind of machine learning algorithms for finding clustering. So that didn't take long at all, couple seconds. Now we're going to go on to actually producing our UMAP. So this is our non-linear dimensional reduction. So these UMAPs are what we're actually going to plot in our app. So this is what we are actually here for. So there's a built-in command to Seurat called run UMAP. We're just going to copy and paste. So that maybe took 10 seconds. And we're not just going to plot it here, but as you can see, you're able to now plot something that looks like this. And this is, because we're following the tutorial, this is the exact same plot that we're going to produce when we actually build our app. But we're not done yet, because we need to assign cell type labels. So at the minute it's just randomly assigned labels to these cells, so numbers from 0 to 8. But we want to actually look at cell labels. We actually want cell labels, and the way we do that is we look at what genes are actually being expressed in each of these clusters. And because it's known, like in science, it's known that certain cell types express certain markers, you can assign cell types, cell labels, based on the gene expression, so based on those markers. For the sake of time, we aren't actually going to do this marker expression ourselves, but I would recommend just going through this and reading through it. It's fairly interesting. But what we're actually going to do, what I'm going to do is I'm going to skip down all the way past this, all the way past the heat map, going to this like assigning cell type identity. So I'm just assuming, you know, you've done the marker analysis, and we've come up with this, like cluster 0 expresses these markers, therefore we think it's this cell type. Cluster 4 expresses this marker, therefore we think it's this cell type, and that's kind of the logic in how we do the cell labeling. And yeah, we're basically going to produce a graph like this. So I'm just going to copy this box here above the UMAP here, copy and paste it into my text editor, but I'm actually going to remove the last two because we don't want to plot it. But I'm just going to run these lines, and those are run now. And I believe that is pretty much it, apart from saving it. So I'm going to copy this save line. We have to change that slightly. But before we save it, I just want to show you what the metadata of this Seurat object looks like. So now we have these Seurat clusters, but if you notice, the cell labels aren't in here yet, so we need to actually add the cell labels in. And to do that, we can just add another line underneath here that says, so to add a metadata column, you just do the object and a $, and then and then what you want this column to be called. So we want to call it cell label. Actually, we'll just call it cell type. This is going to be equal to the idents of the object, because we just renamed the idents to these cluster IDs. So if I run this now and then look at the head, now we see that we have this extra cell type metadata column where for every cell it's given a certain cell type. Now, what I'm actually going to do is just assign some extra data, because in a normal data set, at least in my experience, you're going to have a lot of information, like clinical information about cells. So where the cells came from, how old was the person, what was their sex, what was the disease. Like, you're going to have a lot of extra columns which kind of tell you extra information. And we do want to have some, we're going to assign some random columns just because we want to be able to visualize different aspects of this metadata over the UMaps eventually. We'll get to that eventually, but what I'm going to do is just create some random columns. So bear with me. So underneath here, I'm just going to write a comment, just going to say randomly assign cells, let's say age, sex, and a sample ID, for example. So firstly, for reproducibility purposes, I'm just going to do a set seed, and then let's randomly assign sex to each of these cells in our SIR object. So to do that, we're going to do as follows. So for each cell in our dataset, we're randomly assigning either male or female. So if I just run this, and then we need to run another command, which is actually, we haven't actually set it to our SIR object yet. So we need to basically do pbmc.sex is equal to sex. So that'll actually set it in the SIR object if you run that as well. Now if we look at the head of our SIR object, we now see this extra column called sex, where each cell has randomly been assigned either male or female. So we're going to do a very similar thing. Now let's do age. So we'll just say age, and we'll randomly assign every cell an age between 18 and 65. Just random. It doesn't matter. Whatever you want. I'm going to do it between 18 and 65. So we do round. So we do like that, and then we need to actually add it to the SIR object. So pbmc $age is equal to age. So let's run these two. If we look at the head of our SIR object again, now we see this age column where each cell has been assigned an age. This is not the age of the cell, but it's the age of the patient that the cell came from. And finally, let's just do sample ID. So sample ID Okay, so this is sample ID. Let's run this. We're basically assigning each cell a random sample ID. So sample ID 1, sample ID 2, all the way up to 10. So let's run this. And finally, one more time, we'll look at the head. And yeah, we see every cell now is coming from a sample with a random sample ID. Cool. So that's a good start, and I think that's enough metadata for us for our use case today of like visualizing this data over our metadata UMAP in our app. So the final thing you need to do is save the Sura object, and you want to save it to the same location where you have this folder. So for me, that's in here. I'm basically going to save it in here. So to find the path, you just do, more properties, just copy the path and just save it in here. So this is the path for me. And okay, you'll notice this is a bit weird, but this is another way. Instead of doing double backslashes, you can also do this, which is where you do R. A bit like in Python, you have like a R to escape the special characters. You basically just do this around your Windows path, or you can just do double backslashes. It really doesn't matter. But you're going to save this object. So finally, I'm going to copy and paste that, and this will take 5 or 10 seconds to save, I imagine. And you'll notice that I'm just saving as Sura object.rds. That's important actually. Well, it's not that important, but I'm saving it as an RDS file, which is like a type of RData file. Basically, in our app later, we're only going to be allowed to upload certain file types. So I mean, you can save it as an RDA, or whatever object it will allow you to, but that will kind of impact the development later on. So I just save it like this. So it looks like we're saved. If I go and check the folder, yeah, it looks like it's now been saved in that folder. So that's pretty much it for the actual single cell tutorial stuff. I know I kind of breezed through that, but I didn't want to go too much into detail, but I also didn't want to go too much the other way where I kind of breezed over it too quickly. So hopefully I found a decent balance there. But again, like, if you really want to go through this tutorial and understand it fully, then be my guest. I think that's a good use of time. It's quite a popular thing in bioinformatics these days, a single cell. It seems to be advancing a lot as well. So that's it for the single cell tutorial, and I'll see you in the next section where we start to build out the UI. Of the tutorial, we are going to actually be building out the RShiny app. So what you're going to want to do is open the text editor of choice that you're going to be using. So I use Virtual Studio Code to do my coding, but it doesn't really matter, whatever you use. And what you want to do is you want to open the folder where your Sura object is stored. We're basically just going to be building out in this folder for now, just for ease, so we have everything in the same place. So you just want to open that folder in your text editor. Okay, so I've now opened that folder in the text editor, and as you can see, we've got the Sura object here, and we've got the filtered matrices. This filtered matrices folder, we don't actually need that for the app, but it's just there for the sake of being there. So first thing we're going to want to do is create an R file, and we're going to call it app.r. This is the standard naming convention for naming the file that's going to contain the code for your RShiny app. And as I mentioned before, the structure of any RShiny app contains three main components. You've got the UI, the server, and the function which calls the UI and the server. So the first thing we're going to do is just code out the template of our RShiny app. Okay, so there we go, they're the three main components, and what we also want to do at the top here is install the rshiny package itself. So I already have it installed, and I'm just going to read it in using the library function. However, if you don't have it installed, you need to use the install.packages function and then wrap it in quotes. So actually what I'm going to do is I'm going to open an R terminal alongside this so we can actually run the code at the same time. So I've opened a terminal on the right hand side here alongside the file that we're actually coding in, and I'm actually in the folder where this file is located. So I wonder if I just list the files here, yeah, I should see that file which we're currently coding in, as well as the Sura object, and then this folder containing our matrices prerequisites. So what I'm actually going to do is I'm going to enter R. So I'm just going to type R on the terminal, and now we're in R here, and we're just going to try running library rshiny. So you just want to check you have it installed. Oh look, I don't have it installed. No package called rshiny. And that's actually my error. It's not rshiny, it's just shiny. So remove the R. And yeah, so that's installed. That's installed for me. And if you don't have it installed, you just need to run this command. Install the package shiny, but I've already have it installed, so I don't need to run this. So what I'm going to do to start with, I'm just going to copy and paste everything we have on the left hand side into the terminal. This is not the most optimal way to code, but I just kind of want to show you what's actually happening in the R console when we run these things. Okay, so we're getting an error straight away when we try and load in the UI, and that's that error in dashboard page, da-di-da-di, could not find the function dashboard page. The reason we're getting this error is because the shiny dashboard we're building, it uses its own R package, and that is a shiny dashboard. So I'm just going to paste it in under the library shiny. So as you can see what it looks like, it's this, so library shiny dashboard. So I'm just going to load that in. Again, you might have to install this with install packages. But once you've installed it and loaded it in, we'll try to rerun the UI, and this time no errors. So basically this specific layout of the rshiny UI, it's recognized by this shiny dashboard package, so that's why we need to load that in. Next we're just going to copy and paste our server across, and finally we're going to paste this function shiny app, and as you can see we get this output that says listening on HTTP and then this IP address. And what this is your local IP address. It's kind of your local host. So you might have seen this Chrome icon kind of pop up along the bottom, and what that basically is our app. So if I just bring it into the main screen here, we can see this is the basis of our shiny app. So here we can basically see the subsection. So if I quickly go back to the code, we can see we have a dashboard header, a dashboard sidebar, and a dashboard body. And if we look at the app, we can see these three things. We've got the dashboard header, which is this bar along the top. We've got the dashboard sidebar, which is this part along the side where you can see my face, and then the dashboard body, and this is this main kind of large white space. And the built-in features of this shiny dashboard, you have this kind of toggle feature here where you can close the sidebar by clicking that. So look, we already have the basic layout of our app, and all the code we've written so far is this. So now all we have to kind of do is populate the UI and then code out the server, code out the actual back-end functionality. So the first thing I'm going to show you is with the dashboard header. So as I mentioned, the dashboard header is this part along the top here. So what we're going to do is add a title, and we do that by inside the dashboard header parentheses, we're going to write title equals, and then whatever title you want. So let me just put title for now, and then if we save that and then we refresh our Shiny app, nothing happens. So the reason nothing happened is because, well, as I mentioned, this is not the best way to develop your Shiny app, to be copying and pasting things across. The only reason I showed you this is so you could actually see the errors we're getting, but there is another way to develop. So every change that we make in this file will automatically be updated on the actual app itself every time we refresh the app. So I'm just going to press Ctrl C. That will basically take us out of R. I'm not going to save the workspace, but there's another way to start the R Shiny app, which I'm going to show you now, and you don't actually need to be inside of R to do it. So the other way to start the R Shiny app, and this way is going to allow us to make changes in real time, so that we can make changes, and every time we save our app and then refresh the app, every time we save this file and then refresh the app in the browser, we can see the changes. And the way we do that is using this command. So it's rscript. Well, so firstly you need to move to the directory which contains your app.r file. So we're already in there. It's this R Shiny dashboard. That is my current directory. So you want to do rscript.exe -e, and then in double quotes I want to put, I wonder if I can zoom in here. Hold on. Okay, so I've zoomed in a little bit. I think I clicked enter too early. But yeah, so you want to type rscript.exe -e and then shiny double colon, then run app with a capital A, no spaces, and then in parentheses we want to call our file. So we've called our file app.r, and you want to call your file that as well. That's the command there. So rscript.exe -e and then in quotes shiny run app.r. And if we click enter now, we should see a similar thing where we end up with this listening on HTTP and then this IP address. So what we can do is copy this into our browser. I'm just going to override this, and here we are. So here's our app, and this time you can actually see title has appeared up here, because if you remember, we have this, put this title in our dashboard header, and in theory if we now change this to something else, if we now save this, we just Control S and then we move back to our browser and refresh. Yeah, as you can see, the title's now changed in real time, so we can make changes to our app and then view those changes straight away. And this is really good, especially later on for debugging and making small tweaks to the app. Okay, so what we're going to do now is we're going to add some menu items to our sidebar. So we're going to add two menu items here where you can see my mouse hovering, and this is going to allow us to have a home page and then click a separate menu which is going to take us to our RNA-seq Analyzer app. So let me show you how to do that in the code. So inside your dashboard sidebar function, we are going to add what's called a sidebar menu. So let's, in a new indented tab below, we're going to add a sidebar menu, and we're going to set the ID is equal to tab. So every kind of element we add, we're going to want to add like an ID. So this allows the app to recognize what component we're talking about if we reference it in another part of the app. So I'm going to put ID equals tab, and then beneath this we're going to add two menu items, as I mentioned. So the first is going to be our home page, and the second is going to be our single cell RNA-seq Analyzer. So this is what the structure of a menu item looks like. You have the first argument inside here is a string. So this is what's going to appear over the menu item. So this is what we're actually going to see on the app itself. We're going to see a menu item with the name home page. Now tab name is an ID which is given to the menu item that allows the RShiny app to recognize which component we're talking about. Let's say we want to add a certain plot in the home page menu, or certain text or whatever. We set the tab name to home, so we're not actually going to see this home on the app itself, but behind the scenes the server is going to recognize home. So maybe I didn't explain that well, but we've basically got two IDs. One is kind of the human readable one, the one that we see on the app itself, and the second one is the machine readable one. So this is what the actual app is going to recognize it as. So these are both ways of naming our menu item. It's just one is meant for the actual user to see, and one is meant for actually referencing what this item actually is. I think the tab name is not allowed to have spaces in, whereas menu item can just be whatever you want. The third parameter we've had here is an icon. So if I just save this and reload it, you can see what's happened. Yeah, so icon. So here you can see three sort of rows here next to the home page, and that's our icon. There are different types of icons, but we've just put like list here, and that's what's giving us that there's 3 lines. So I can zoom in. Okay, yeah, that's quite cool. So if you zoom in, the app will actually react. So it's got a lot of kind of CSS and JavaScript behind the scenes, which is really great because we don't really have to do any of that ourselves. We can add our own CSS and JavaScript if you want to add some extra features, but the cool thing about RShiny is that a lot of that's just built in. So a lot of this interactivity, this like dynamic content already built in, which is great. Okay. You can actually get rid of this. Well, you don't want to X out of this R script, but I'm just going to move out of the way because we don't really need to interact with this at all anymore. We just need to make sure that's running for our changes to be seen in real time. So I'm just going to move it down here. I'm basically just going to hide it. I can move my face over this side. Okay, so now we're going to add a second menu item, and when we do that we want to put a comma after the first one, and then pretty much type the same kind of structure for this one, but we're going to name it something different. So here, yeah, exactly the same thing. We've got the name, which the user is going to see. We've got the kind of ID, which references this certain menu item, and then we're adding an icon. And again, icon is like a built-in feature of RShiny. We're calling this one's called Edit, and I'll just show you what that looks like. If we refresh our app, it kind of looks like this. It's like a pen within a box. And what we can do is now we can switch between these, and when we click on them, the kind of background color will change, and there'll be this kind of strip on the left here that lets us know which ones we've selected. And even if you just hover over one of them, it will kind of dim to show you that you're hovering over it. Again, all kind of built into this RShiny, which is great. Okay. So with our app, what we want to do is on this home page, it display like a welcome message. And when we click on the single cell RNA-seq analyzer menu item, we want some things to appear beneath it. We want a file upload and a run experiment and a reset experiment button to appear when we've clicked this. So that's what we're going to work on now. Because those are going to be in the sidebar, what we're going to do is add another comma after the second menu item, and we're going to add what's called a conditional panel. Now a conditional panel is something that's something in our sidebar menu that's only going to appear when a certain condition is met. And in our case, we only want certain items to appear when we've selected the single cell RNA-seq analyzer menu item. So to do that, we use a conditional panel, and we need to set a condition as the first parameter. And our condition is going to be that the menu item selected is single cell RNA-seq analyzer. So let me show you what that looks like. So here we're saying condition equals input.tab equals input. Now that might be a bit confusing because input, as I mentioned, is an inbuilt R kind of term. So in the server down here you can see we've got this input. So this first input, input.tab, is kind of relating to that. It's basically saying the input, which is our UI, the tab ID, so whatever tab is selected, if that tab selected is input, and in our case, remember the menu item, we're setting the ID of this second menu item to input. So when the tab selected is the tab with the ID of input, then we want to perform some condition beneath here. So let me just code out the rest of this and I can explain kind of what's going on. Okay. I'm actually going to add another tab here just so we can see where the sidebar menu starts and finishes. But basically, when this condition is met, so for the conditional panel function, the first parameter is your condition. The second parameter, so you put a comma, and then the second parameter is what happens when this condition is met. So in our case, when this condition is met, we're adding a div element. So if you know anything about HTML, a div element is just a block of code, if you will, that's going to be input right below the menu item. So within this div item, we have three things. The first one is a file input. Now the file input, again, is a built-in functionality of RShiny. It allows you to upload a file, and as you can see, there are 4 parameters we're specifying. The first is the name. So again, this is what the user is going to see. The user is going to see, so the first one is the ID. So this is what RShiny is going to recognize this element as, so it's file. the second one is what the user is going to see this time. So upload file, that's what's going to appear on our app. Multiple equals true, so that you can set, this is an argument, you can set it true or false. If it's true, it allows you to upload multiple files. If it's false, it will only allow you to upload one file. So we actually want this to false, I just realized. And then finally, what file extensions are we going to accept? Now here I've put .rds only, but this is a vector in R, and you can add other file types. So let's say you wanted to allow CSV files and other types of files, you can add them all into this vector here. But we, if you remember, our Surrey object has got the .rds extension, so we're just going to put it as .rds for now. But if you saved it as maybe .rda or some other R file type, feel free to add your extension into this vector here. Now, beneath this file input, we have two action buttons, and before I explain these, I'm actually just going to save it and show you what it looks like now. So if I refresh the app, if we click on the single cell RNA-seq analyzer, we have the upload file, and then we have the two buttons, and these don't do anything. Well, the upload file button will, and you can upload the file, but the buttons don't actually do anything. And you'll also notice maximum upload size exceeded, and that's an important error which I'll show you how to fix. So basically, by default, you can only upload files of a certain size to R, but you can change that manually. You can just add something in your R script which will allow you to accept pretty much any size of file you want, but you want to kind of, well, yeah, whatever size of file you want, to be honest. So take a look at this. We've got the file, and then beneath it we've got two buttons. So if we just go back to the code, I can explain to you what's going on here. So the buttons, these are created using the action button function. Again, this is something which is built into our Shiny. Now the first argument is the ID. So on the back end, this is what our Shiny recognizes this as. So this action button has the ID of reset, this action button has the ID of run, and then the next argument is what is actually shown on the front end. So reset and run are shown on the front end, actually on the buttons themselves. You'll also notice on these buttons there's symbols, there's icons. So this one's got a kind of reset icon, this one's got like a play icon for run, and that is our third parameter in each of these. We've got icon, just like we do for the menu items themselves. We've got the icon equals icon undo, and then icon equals icon play for those buttons respectively. Now the last piece of the action buttons here, this is some custom styling in CSS. So in fact, let me just remove these just to see what happens if we remove them. But basically, as I said, you can add your own custom CSS and JavaScript into the R Shiny to add different like styling or different dynamic features. So without those, what happens? So without those, one, they're not colored, so we've basically added our own custom coloring. A second thing is that they are of different lengths, so we are basically setting them both to be the same length. Let me just show you. Let me just undo. So first thing is color. We're adding color. So it's a text color and then a background color, and then what we're doing is adding a width. So the width in both cases is a percentage of the whole width of the sidebar. So 87.25, it was just kind of a num. It just looked, there's no like reason in particular I picked that number, but it just looked about right on the screen. So you can play around with different colors, you can play around with different button widths. If you do not set a width argument, the width will just be pretty much as long as the text is. So in our case, run is always going to be shorter than the reset, unless we set the width icon to make them the same. But that's it. So all we have done so far is built out a little bit of this UI function, and we've already got an app which is, if we just refresh again, an app which is looking pretty good. I'll just change the title to something more relevant. I'm going to call it single cell RNA-seq analysis. So if we save it and refresh, there we go. That looks good, very good. Okay, so next we're going to fill out this dashboard body. So the dashboard body, as I mentioned, is this large white space which pretty much takes up most of our app, and this is where we're going to display some instructions, and we're also going to display some results eventually once we click the run button. So to start with though, we're just going to have a single tab with some basic instructions on how to use our app. So what appears in your dashboard body depends on what menu item you select. So if we select home page, for example, we want something to come up here, whereas if we select single cell RNA-seq analyzer, we want something else, something different to come up in the main panel. So bear that in mind, because when we start filling out this dashboard body, we need to add what are called tab items. So again, this is built into our Shiny, and here we're going to list 2 tab items. So we have 2 menu items, and we want to basically set 2 different main pages depending on what menu item we've clicked on. So within this tab items function, we are going to add tab item, and we're actually going to add 2 of these separated by a column. So in each of these, we're going to build out what we're going to see in the main panel with when each of our tabs are clicked. So again, I've kind of banged on a lot about how we have these like IDs. So each element, like each menu item, has a tab name, and each kind of file input has an ID. And the reason that that's important is so we can reference those elements, those menu items, those buttons, whatever it is. We can reference those in other parts of our app. So for example, here in the tab item, we want to, this first one, we're going to reference this menu item. So this is going to be the content that's displayed when we click this menu item, and we do that using this tab name again. So we can say tab name, just like we have here, tab name equals home, and tab name equals input. We can just copy those into here. So tab item, where tab name is home, we can add some functionality or some display within this tab item. So within this page, we actually want some sub-tabs. Like we want one instructions tab, and then once we run the experiment, we want some other sub-tabs displaying the results to appear. So to do this, we're going to use a tab set panel. And again, we're going to provide this an ID so we can reference it in other parts of our app. So a tab set panel is basically a panel which can accept multiple tabs. So as I mentioned, to start with, we're just going to have a single tab, which is going to be our instructions. So what I'm going to do is just create that single tab. So we're going to say tab panel, we're going to call it instructions, and here we can basically write anything we want. So for example, let's just say hello, this is a tab. If we save that and then see what it does. Oh, an error has occurred, need tab name. Okay, let's go back. Okay, yeah, so we kind of initialized this empty tab item, but it has to have a tab name by default, so let's just add that. Basically that's going to be our input tab, and we'll just save that and run it. Okay, so as you can see now in our home tab we've got the instructions, hello, this is a tab, but actually we don't want it in our home page, we want it in our single cell RNA-seq analyzer. So actually I'm just going to swap these around, so that can be input and that can be home. If I save it and refresh, yeah, now it's only when I click single cell RNA-seq analyzer where this tab appears. And the idea is, like, when we click on experiment, we're going to have some other tabs that appear here, so we can click through the different results, the different figures, and we can also come back to the instructions tab if we want. And as you can see, this text it doesn't look very nice, it's just been like pasted underneath, but we can add a different styling to this. But before we do that, I do want to add some content to our home page. So if we go back here, we basically want to populate what's inside this tab item. So in here we're going to use some actual HTML. So we're going to do something. So if you're used to HTML, you have like headings, like heading 1, heading 2, heading 3, or you can have paragraphs. And you've probably seen like HTML, it uses, you know, kind of blocks of code like this, where you have these kind of arrows and you kind of enclose text within these. So if I wanted to do a paragraph, for example, it would look something like this, where I'd have an open tag, a closing tag, and then within that I'd have some text. And RShiny has kind of its own way of creating those without actually having to write tags. So basically you do tags $h1 instead of writing h1. It's just like it's just a way of interacting with HTML in RShiny that I think makes it easier. And what we do here is we add some parentheses, and then we can add some HTML where we just write something here. So let's say we said, welcome to our app, save and refresh the app. Yeah, so that text is now appearing on our home page in our main panel. But using this HTML function, you can also add HTML within this thing itself. So we could say, you know, underline this using the U tags. So if we save and run that, now it's underlined. So you can set your welcome page to say whatever you want. I think I'm just going to keep it like that for now. I might just say welcome to our Shiny app. So yeah, I've just changed it to welcome to the single cell RNA-seq Seurat analysis RShiny app, and you can see that there like that. I'm just going to keep it like that for now. I think that's fine. Okay, actually I'm going to show you something, a better way of having kind of text within this tab, because at the minute that doesn't look very good. And there's quite a cool way we can add markdown into our RShiny app. So what you're going to want to do is go back to your back into your text editor, and we're going to create a markdown file in this folder. We're actually going to create a folder called markdown, and then we're going to add this file within that folder because we want to keep things tidy, and there may be multiple markdown files you want to use in your app. So I recommend just having folders for groups of files that are going to be similar. So in here we're going to add a file. We're going to call it instructions.md. MD is the file extension for a markdown file. So once you've made that file, we're going to go in here and actually write some markdown instructions for our app. So let me give you some basic markdown, a basic markdown tutorial while I'm here. So for a heading, you use the hash key. Let's say this is heading 1, so heading 1, and then you can have like heading 2, which is 2 hash keys, heading 3, which is 3 hash keys, and this basically these will kind of render as different sizes. Heading 1 is always the biggest, heading 2 is the second biggest, heading 3 is obviously going to be the third biggest. So let's say we want to include this text as our instructions. So we go back to our app, and instead of saying hello, this is a tab, so at the minute, if you remember, we just have hello, this is a tab. So instead of that, we are going to write, we are going to type I'll set it on a new line. We're going to use a function called include markdown. And then parentheses, and then in here we are basically pointing to the file that we just created, so that instructions.md markdown file. Now we can actually use relative paths because that file is also in this directory. We don't have to use the full path. We can use the path that's relative to the file we're in, so the path that's relative to app.r. And so in my case that's going to be markdown.instructions.md. So this is basically pointing to the same directory as app, but then we go into markdown and then instructions.md. So if I save this now and refresh the app, here we can see that we have the 3 headings, so heading 1, heading 2, heading 3. So we've basically, and this is a good way to keep your app tidy, we've basically taken some text that we're going to put in our app and we've abstracted it from the actual app itself, because we don't want to, let's say we had to write, a whole page of instructions. We wouldn't want to have a whole block of text just in our app, just cluttering things, because it makes it overcrowded and hard to kind of see what you're doing. So it's good practice to extract these types of things into markdown files. Now we don't actually want to just put heading, 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 so I'm just going to fill this out with some actual content. So I'm just going to copy and paste what I've previously written, but I'm just going to take you through this. So how to use the app, that's heading 3, so remember the £3 is the heading, it's like the third biggest. And then there's basically 3 steps. Upload a surrogate object, and we're going to kind of go through these steps as we go through the tutorial. But then second step is press the run button to run the analysis, and then we've got kind of an indent here, and then the third is press reset button. Now you probably noticed some funky stuff going on here, like what are these stars surrounding upload file, and what are these backticks, triple backticks surrounding run and reset. Well, I think it's better I just show you rather than explain it. So let me refresh. So upload file is now in bold, so these double stars around the phrase, that makes it in bold, and the run and reset are now in kind of a different color. It's almost like a code, or it's kind of like, you know, it's a different background color. It allows it to be easily visualized. It's almost like this is a command rather than just a normal word. So the 3 backticks has this kind of dimmed red color. So that's, I'm just going to leave it as that, quite basic instructions, but I think that's a kind of good way to demonstrate how to use the markdown file. I'm just going to save it and exit out of that for now. Okay, so that is the bulk of our UI built. If we go in here, you can basically see that's everything, and if you want to have a little play around with this, you can. So we've basically got a sidebar which we can minimize. We've got a file upload button which we can click. And actually, something I'll just point out here is in our code we did set like the file extensions which our app will accept, and so here we've just got RDS. And what you'll notice is when we actually do try and import a file, in here, in this bottom part here, we see this is kind of pre-selected RDS. So that's basically what that means. But what you can do is kind of ignore that, and you can just say, okay, I just want like all files. I don't just want RDS. I want all files, and so you can do that as well. But it's just an extra feature I wanted to show you guys. And actually, let's just try and upload a file that isn't an RDS file. So it does work, so it doesn't throw an error if you upload a file that isn't one of the accepted files. But then what we're going to do is we're also going to validate that our file is a Sura object and it is an RDS file in our backend, in our server function. So now let's get into building out the server function. So at the top here we are going to add an option which allows us to import large files, because something I didn't actually show you was that if we by default just try and upload this Sura object, we get this error, this maximum upload size exceeded. And the reason we get that error is because by default Shiny limits the file upload size to 5 megabytes per file. But if we look at this Sura object, we can see it's almost 300 megabytes. So it's 287 and a bit megabytes. So we basically want to increase the default file upload size per file in our Shiny app set. And the way we do that is using this command. So we type options shiny.max request size and we're going to set that equal to 300 megabytes. So our Sura object was 287 megabytes, so we're going to set it to 300 megabytes by doing this. So I'm just going to save that and refresh the app, and let's see if our warning is removed now once we upload the same Sura object. Yeah, so this time we can see it's taking a lot longer, and for larger files it will take a long time to upload. Like our Sura object isn't actually that big, only 300 megabytes, but it does take, you know, a good sort of 5 seconds to upload. But as you can see, the max file exceeded has been removed, and we get this upload complete. So that's good. Now the next thing we're going to do is we're going to disable the run button. And what I mean by that is, by default, run is clickable. So if I just refresh, we can click run. We only want to be able to click run when we've uploaded a file. So we're going to disable it by default and add some logic where once a file's been uploaded, then this button becomes clickable. And to do that, we need to use something called Shiny.js. So we're going to go back to the top of our app, and we are going to import this package called Shiny.js. So you just use library. Again, if you haven't got it installed, you need to install it with install.packages. And what we're also going to do is we need to put use Shiny.js in our UI. So we can put it anywhere in the UI. I'm just going to put it in the sidebar menu here, because we're going to be using the run buttons in our sidebar. So I'm just going to put use Shiny.js. But the fact we've got that there means any element in our app, in our UI, can be disabled or enabled dynamically with the JavaScript. So now in our server, what we need to type is Shiny.js disable, and then the ID of the button that we want to disable. So in our case, we want to disable our run button, and I believe it's lowercase run for the ID. Yeah, so our ID lowercase run. So if you remember, this is the ID and this is the actual what appears on the app, the actual what the user sees. So we disable run by default using Shiny disable run. Again, I'm just going to quickly save it, refresh the app, and we've got an error. So error sourcing. What's going on there? Okay, so I realized I forgot to put a comma after the use Shiny.js. So just make sure you put a comma there. If we save that and rerun it, yeah, so that's working now. And if we go to our single cell RNA-seq analyzer menu tab, we can see the run button has been disabled by default. So when we hover over it, we get this kind of our cursor turns into this kind of symbol which represents us not being able to click, whereas if we hover over the reset button, our cursor turns into like a hand pointing. So that means the run button is disabled now by default. And what we want to have happen is once we've uploaded a file, we want the run button to become clickable. So just like the reset button, we want, when we hover over it, we want it to be a hand, and we want it to be its normal color, because as you can see, it's kind of like dimmed now. So if we just wait for the Sura object to be uploaded, it shouldn't change the fact that this button is dimmable, is unclickable. So upload complete, yeah, and the button's still unclickable. So we need to add some logic there to change that. So in our server function, we can use something called an observe. So we can observe an event, and this is basically relating to the input. So when a certain, as I mentioned, our server function here has an input and an output. We can observe for when a certain input occurs, we perform some logic. So for us, when a certain file is uploaded, that's an input, we want to enable our run button. So let me show you how to do that. So we're going to use something called observe. So here we're just going to use a simple if-else statement to check if the file exists. So if the file has been uploaded, enable the button; if not, disable the button. So simple if statement in R, if is.null input $file. So I'm going to explain this in a second, but let me just clear out the rest of this. Okay, so let me explain what's going on here. So is null input $file is checking if there's a file uploaded. So this file path, what's that referencing? So as I mentioned, every component we have in our UI, we have to have like an ID. So for this file input, our ID is file. And so what that means is when we upload a file, it's given this ID. So it's kind of like a variable in R. It's given, this is its variable name. So then we can play, in fact this is not its variable name, its variable name is input.file. So anything, it's basically being added to this like general input parameter that's our server. So any input to our UI, whether it's a file upload, whether it's a button click, whatever it is, it's going to be labeled as input $, the ID of the thing. So in our case, input $file, because it's a file upload. So we're saying is null input $file, and then if it's not equal to true. So if our file, okay, so this is kind of like a double logic going on here, but we're saying, so like if is null is not true, which means like if the file upload is true, we're going to enable our run button, else disable. So we're disabled by default. If there's a file being uploaded, we want to enable it, and then conversely, if we remove that file and there's no longer a file uploaded, we want to disable it. So this observe function, it's kind of always looking out for changes to this variable, input $file. So I'm just going to save this and refresh the app, and as you can see, by default our run button is disabled. But if we upload a file, and I'm not going to upload the server object just because it takes a while, but let's just say any file, if we upload the file, we then see the run button becomes enabled. So we can now click this button. It doesn't do anything yet because we've not hooked it up to any functionality yet, but it becomes enabled. And I'm guessing if we remove the file, which I don't think we can, I don't think we can, but if we remove the file, it will become disabled again. But we're gonna basically, the next thing we're gonna do is when we click this reset button, we want to remove that file. So let's do that now. And then this time, instead of using observe, we're going to use observe event. So it's very similar to observe in the fact that it's a reactive value. So it's always on the lookout for changes in a certain variable. So let me just code it out and I can explain it. So we're going to say observe event. This time our event, instead of being input $file, it's going to be the button click. So what is our reset button called? We called it reset. So we're going to say on input $reset, and then we're going to have some logic. So on observing the event input $reset, we're going to have something happen. So when we click reset, we want the file that's been uploaded to be removed. So to do that in our Shiny, we can use the Shiny.js again. So we say Shiny.js reset file. File again, that's the input ID to our file upload. And when we remove the file, we also want our run button to become disabled again, because our button the run button should only be enabled when there's a file present. So we're going to also put Shiny.js disable run. Now let's see what happens here. So there seems to be an error here, because as you can see it's kind of dimmed out, so this means there's an error. What's going on? So let me open the terminal. Could not find function observe event. Okay, so it looks like I've just made a spelling mistake. It's not observer event, it should be just observe event. So let's correct that, save and refresh, and yeah, so now it's working. So by default run button's disabled. if we upload a file, let's find a small file, it becomes enabled, and if we click reset, the file is removed and it becomes disabled again. Cool, so that's the functionality of our run button sorted. Okay, so that's basically a lot of the kind of simple functionality of our app. Like, you can picture, like, this is the kind of thing you can have in any app. You can upload a file, click a button, whatever. Now we're going to get into the specifics of the single cell RNA sequencing. So when we click this run button and we've uploaded a surrogate object, we want it to make some UMAP plots for us and to plot certain metadata or certain gene expression over these UMAPs. And so this is a lot of back-end functionality which we need to kind of code out, which is going to basically, we're going to have a lot of built-in surrogate functions. So surrogate functions for creating UMAPs or feature plots. These are two types of plots for viewing our data. We are going to be using these on the back end. So it's best practice in R Shiny to have a separate file for all your back-end code. So for any processing that's going to be going on when we click a button, we kind of going to have all that going on in the back end. So to do that, you want to create a new file in the kind of root directory, in the same directory as app.r. Create a new file and you're going to call it global.r. And this is actually best practice, and so what we actually do is we contain all our back-end code in here. So what I'm actually going to do is go into our app.r, and any package installations that we have so far, we're just going to get rid of these. We're going to cut them and we're going to paste them into global.r. And then what we're going to do is we're going to go back into R and we're going to do source global.r. So what this is doing is it's reading everything inside global into this file. So instead of loading the packages in here and then running this code, we're loading the packages into global.r and then we're loading global.r into our app file. And that seems like quite complicated right now. Like, why would we just like read it into here? Why couldn't we just read it straight into here? But the reason for that is because we're going to have a lot of extra code, like back-end processing code, in this global file, which will probably use a lot of these packages as well. And it's good to just have your sort of any functions or any packages you want to import, have them all kind of in one place. So that's what this file is for. So let's think about this. So what do we want our app to do? First thing, when we click this run button and we have uploaded a file, we want to read in the file. So we want to read in this surrogate object. In fact, before we do that, we first want to check that it is a surrogate object, because you could just upload any file and then try to read it in, and if it's not the file that like it's expecting, it might throw an error. So we need to add some functionality that's going to validate that our file is a surrogate object. And we are also uploading an RDS file, so we're going to validate two things. One, that it has the RDS extension, and then secondly, that it is a surrogate object. So let's do that in our global file now. So we are going to create a function to do this. We're going to call it load surrogate object. We'll just call it obj. It's going to be a function, and it takes a path. So basically, it doesn't take a path. It takes the file that we upload. But the file that we upload, if we actually take a look at it, and you know what? Let's take a look at it. That's quite an important thing to show you guys. So I'm going to go back to here, and let's. Where are we? So in our server, observe. Okay, so when our file has been uploaded, we're just going to print out. Let's just do print out input $file, because this is quite interesting. So let's just refresh our app. Oh, we've got an error. Yeah, sorry. So I've kind of started writing this function. Let me just code it, comment it out for now. Right, so when we upload a file, for example, in fact, let me just pick a small file. If we upload this, it should have printed that out. Now let's have a look. Okay, yeah, so it's printed out input $file. So it's actually a path, but it's not the path to our file. It's a path to a temporary file. So it basically takes the file we upload and it kind of copies it to a temporary location. So as you can see, this kind of temp folder, and so we're now going to like read in this file. So let's get back to our load Sura obj function. So it's going to take that path, which is the input of a file, and what I want this function to do is one of two things. Either it's going to validate that the file is in the correct format and that it's a Sura object, and then it's going to return, the Sura object, or it's going to throw an error. So we're going to do some error checking. We can use like try catch in R. Like if this file is not in the format we expect it to be, we want to throw an error. So this is what we're going to code out. So I'm going to initialize a vector just to store the errors, because we basically want to catch different errors and then feed that back to the user, so the user knows why their file didn't get accepted. So we're going to say errors empty vector. Next thing we do is check the file extension. So we're going to say if, so we are expecting the file extension to be .rds. So we're going to check that it is .rds, and the thing is, I think it can be capital RDS or lowercase RDS. I don't think it matters, so we want to ignore case. So let's do if not to lower, and we're going to use this package called tools. So I'm going to add another package here at the top. Library tools, if not to lower tools file ext. So what this does, it basically just checks the file extension of a path, because remember, our input is the path, and then I need to put RDS. Okay, so this is, if our file extension is not RDS and it says to lower, so basically any case RDS, it could be all caps, all lowercase, different like camel case, like if it's not RDS at the end of the file, then we want to add an error to our errors vector. So we're just going to say we're going to add the error invalid RDS file to our errors vector. Okay, so the next thing we want to check, if it is an RDS file, we actually want to try and read in the file. So try to read in file. We're going to use a try catch block. And we're going to try and read in the file using the read RDS function. So read RDS is a built-in function in R for reading RDS files. It's like this, so read RDS. And we're going to put our path in here. So we're going to try and read our Sura object into a variable called obj. And then if there is an error, then we are going to add that error to our errors vector. So error equals So we're actually going to add the same error. So if we can't read the file in with read RDS, we're going to throw the same error, invalid RDS file. And then return the errors. So the idea with this is we're basically passing checks. If we pass the file extension check, we carry on. If not, we stop the function and we return an error. And the idea is the user is going to see what this error is. So that's why we're adding an actual sentence with what the error is to this vector, because we want to return this and then display it to the user on the app itself. Okay, so we've checked the file extension, we've tried to read in the file. If both of those things pass, we want to do one more thing, and that's validate that it's a Sura object. And to do this in R, we can use the inherits function. So built into R, it allows us to test the kind of type of an object. So we're going to do if not inherits obj is of type Surat, then we're going to add another error to R. We're going to say errors equals errors. File is not a Sura object. And then also, like, we can return the errors again if it throws an error. Okay, so basically check 1, check 2, check 3, and if it passes all 3 checks, we just want it to return the object, return the Sura object. So that's our first backend function. And as you can probably imagine, with large R Shiny apps, you can have a lot of kind of backend functions, a lot of like stuff going on, which is why it's good to separate out the actual app stuff to the kind of backend processing, because we don't want this to get too cluttered. Okay, I think we should test this function out, just so we can see what the heck was going on and see if it actually works. So I'm just going to create. I'm just going to open a new terminal. If I just do so, I'm just going to open up a command prompt. I'm just going to move it over here for now. Oh, what's going on? And so I'm just going to enter R and we just want to check that this function is doing what we want it to do. So I'm basically just going to copy and paste everything into here. I mean, you don't have to do this, but you can just watch me do it. So we've loaded our function, no errors so far, and we basically want to pass a path into this function. So let's just call the variable out equals, I'm going to call our function load Surat obj, and we need to pass the path to our object. I'm trying to think which directory am I in. Okay, I'm just going to run that. I'm just going to check my directory first. Okay, so I'm in this directory. I'm in the R Shiny directory. What I need to do is pass the file name, because it should know the file is in this current directory. So Surat object.rds. So that's the file name I'm getting from over here. And yeah, as we can see, it's loading. To load in the Surat object, it will take 5 seconds. Okay, so that took about 10 seconds, but we can kind of see this is a Surat object. So it's worked. Great. But now let's try and test if we try and put a file path that's not a Surat object. So let's just take, for example, the app.r file. If we try and load this file as a Surat object, as you can see, there's no loading time, and what I suspect has happened is it saved an error. Yeah, so invalid RDS file. So basically what it's tried to do is check the file extension. It's realized it's not an RDS file extension, so it's added the error invalid RDS file to this errors vector, and then it's returned the errors vector, and that's what we have here. So the function works. It's great. I'm gonna close out of R, and we can get back to the coding. So so the first thing we're going to do is click this run button. We're going to load in our Sura object, and we're going to validate that it is in fact a Sura object. So we've done that. Then what we want to do is display some plots. So for the sake of this video not being too long, I know it's already going to be quite long, but we're going to assume that our Sura object has the UMaps, like the UMap kind of field already contained in it. So within Sura, we went through the tutorial, and you run the run UMap command on your Sura object, and it creates this kind of data field in your Sura object. And we're going to assume that we're not going to perform a validation on our Sura object to check it has the UMap field. We're just going to assume it already does, because at least we know it does in this example. So the next function we're going to create is one called, well, we need two. One is going to create the UMap that's going to display our metadata, and the other is going to create a UMap that's going to display gene expression. So let's create two. The first one's going to be create metadata UMap. It's going to be a function, and the function is going to take the Sura object and the column, the metadata column that we want to display over the Sura object. Okay, now I've kind of developed some custom functions for plotting these UMaps. So what I'm going to do is I'm just going to paste those into here, and then I'm going to talk you through it. So there's a lot that I've just pasted here, but bear with me while we kind of go through this. So okay, so we have various columns in our UMap metadata. So these are the things that we're going to be plotting over our UMap. So we have things like clinical information, so maybe a patient's age, maybe their name, or whatever. But we also have like quantitative columns that contain like the number of counts, number of features, and the percentage of mitochondrial genes. So we have these things, this information per cell. And so this is a very custom UMap plot. So I'm basically saying if the column is one of these, so these quantitative ones, then I'm going to create a specific type of plot. It's a ggplot, and it's basically going to pass the UMap cell embeddings from the Sura object, and it's going to take the metadata values from the Sura object metadata, and it's going to use those to plot a certain type of map, UMap that's going to be different from like the standard UMap you see using ggplot. The reason I do this is because, okay, so what you normally do is just this, a simple line of code. You take your Sura object here, and you take your metadata column here, and you just plot that, and then you can, that's effectively a ggplot, you can just save it. However, I found like with these quantitative things, number of counts, number of features, whatever, the plot doesn't look that nice. So I've basically made this custom function. You can just copy this straight from my GitHub. Like, I'm probably not going to bother explaining it just because I don't want to dwell too much on this. But this just makes it look nicer for these quantitative UMap measurements. So let me just talk about, okay, high level what's going on. So if the column is one of these, create this UMap. If the column is any other column, create this UMap, right? And if the column doesn't exist, you want to create basically a grey square which just says column doesn't exist. That's all this function is doing. Okay, so that's our create metadata UMAP function. We want to create another one which is like for gene expression UMAP. So I'm just going to call it create feature plot. So in our Seurat object, genes and features, they're the same thing. So create a feature plot or a gene expression plot, it's kind of the same thing. So again, this time we're going to take the Seurat object and a gene this time. Okay, and again, I don't want to dwell too much on the actual function itself. I'm just going to copy and paste from my notes. But at a high level, this is what it's doing. So if you can kind of, okay, let's ignore this for a second. If the gene is in the row names of my object, so in the Seurat object, your genes are your row names. So if the gene is in there, you just want to create a feature plot. This is a built-in function of Seurat, which reminds me we need to load in Seurat. So do library Seurat as a capital S, I believe, like that. You can install it with install.packages if you don't have it installed already. So but yeah, basically saying we create this feature plot. This is a built-in function of Seurat. If the gene exists in the object. If it doesn't, again, create just a grey blank square saying gene doesn't exist. That's all that's going on there. Okay. So this is our global file done, pretty much. Three things. We're going to load in the Seurat object and we're going to validate that it is a Seurat object. If it's not, we're going to throw an error explaining why, explaining like what went wrong. And then two other functions, both to create the plots. So if you remember in our app, when we click run, we can have two extra tabs that appear, and they're each going to show a different plot. And so that's what those two functions do. They create those plots in those two tabs respectively. Right, that was a lot, and that might be a bit confusing, but don't worry, like we're almost done now. All we have to do now is write the code that's going to figure out what's going to happen once we click this run button. So we're going to go back to our app.r file, and as I've shown you before, we have these observe functions. So these are looking out for a change in a variable. So we're going to do something very similar. It's like we're going to observe when the run button is clicked, and when the run button is clicked, we want to create the two new tabs, and we also want to then display our plots in these tabs. Now the plots are going to be created using the two functions that we've just written. So we're going to use another observe event to observe the clicking of our run button. So just like we've got here for reset, in fact I'll just copy and paste it. We're going to observe our run. We're going to observe run. Okay. So when we click run, the first thing I want to happen is that the run button is disabled, because you don't want the person to be able to click it again once they've just clicked it, because otherwise it's going to mess up the processing. So to do that, we just copy from what we've got above. We're going to disable our run button once it's been clicked, just by default. Now what we're going to do is as you saw before, the Seurat object takes a little while to load, 5 or 10 seconds. So we don't want there just to be a static screen, because the user is like, what's going on? I've clicked run, why is nothing happening? We want to show like a loading widget, so like a spinner that's going to say loading. So to do that, we can use another R package. So this R package is called Shiny Busy. Now, as I mentioned, we don't want to put our packages in this file anymore. We want to put all our package kind of imports in this one. So this is Shiny Busy, all lowercase, no spaces. This is what we're going to use to have that loading widget. So paste that into your global.r file, save it, and then come back to app.r into the observe event of our input.run, and here we are going to add our loading widget. So you basically have like a start widget and stop widget. So we're going to put a start widget here on this line, do some computation beneath it, and then once the computation is done, we can do a stop widget. So what does that look like? So we have to do a show modal spinner, and there are different types of kind of spinners and whatnot you can have, but I'm just going to have a basic one. So we're going to use show modal spinner. So yeah, as I mentioned, they don't have to be spinners either. You can get like orbs, you can get like all these different kind of cool loading widgets. You can kind of search them yourself. If you just search like shiny busy loading widgets, you can have a play around with different ones, but we're going to use the modal spinner. So show modal spinner, and then it takes one parameter, which is the text. So we're just going to say preparing plots. Right, let's test that. See if that works. It should just run forever because we haven't actually added a stop widget. But if we click, oh no, we need to refresh first. Oh, we've got an error. So unexpected create UMAP function. Okay, so that's our create UMAP function in global.r. Create metadata UMAP. Oh, it looks like I just spelt function wrong. Okay. Right, let's try that. Okay, so that's loaded. Right, so we're just going to upload a random file, and we're going to click run, and we should see our loading widget. Yeah, there we go, preparing plot. So that's our text argument we've added to the widget. And this is in fact an orb. It's not like a spinner, but you can add spinners or whatever if you just look up shiny busy. Now this will just run forever and ever because we haven't told it to stop, but we can just refresh our app and we're back to normal. But okay, let's kind of keep coding out the app. So we're in this observe event, we're showing our spinner, and then we want to actually load in our Sura object. So here is where we use our load Sura object function, which we've put into global.r. So we're going to say obj, we're going to assign the Sura object to variable obj, load Sura object. Okay, so it already remembers my function, load Sura obj, and then input $file $data path. Okay, so you're probably thinking like, $data path, what the hell is that? Okay, so well, yeah, what is the data path? So actually when you load in just input $file, it doesn't just return the path. Let me show you. It actually returns 4 different fields. It returns a data frame. The first is the name, you got like the name, the size of the file, the type of the file, and then the data path. So we just want the actual data path, so we reference that using this $. So input $file returns a data frame, but we want a certain element of the data frame called data path, which is the actual path we want to load in. So that's what that means, but let's carry on. So we're going to load in our Sura object. Now, as we've seen, this can do one of two things. Either it can actually validate that it's actually a Sura object and load it in, or it can throw an error. So our obj variable is either going to be a Sura object, or it's going to be an error. So basically we want to check which one it is. If it is a Sura object, carry on and render our plots. If it throws an error, we want to display that error back to the user. So how are we going to do that? Well, if we just quickly look at our global.r function, the errors is a vector, whereas a Sura object is not a vector. So what I'm just going to do, I'm just going to do a simple check, like if obj is a vector, then we can return. So then we can return the error back to the user. So I'm just going to say, like, if is vector obj, if it is a vector, that means it's thrown an error. Then we're going to do something called show modal. So this is basically a modal is a pop-up on the app that says to the user a message, and they can click off, they can click like OK to get rid of the message. So if there's an error in loading the Surrey object, we want there to be a pop-up on the app that's going to tell the user there's an error and what the error is. So if it's a vector, so if there's an error, effectively we're going to do show modal, and we need to add some text. We can do that with modal dialog. And here we can add two things, so a title, which will just be, you know, error with file, like a general message, error with file, and then beneath that, separated by a comma, we're going to add the actual text, which is going to display the actual specifics of what the error is. Because remember, the error can be, well, one of two things. One, it's not an RDS file, and one, it's not a Surrey object. So we're going to say HTML, and we're just going to say there was an error with the file and what exactly the error was. So this is how we do that. So H5, these are HTML tags. It's a heading, so heading 5 is the 5th biggest, so it's actually quite small, and it's saying there is an error with the file uploaded, please see below for more details. BR is a break, so basically you start a new line, and then it's going to paste the contents of obj, and it's going to separate that by new lines again. So remember, if this is triggered, that means obj is an error, it's a vector, so it's basically returned the error is vector, so it's either returned invalid RDS file or file is not a Surrey object. And if that's the case, we're going to basically paste that onto our modal. Now this might be a bit confusing. Let me just show you. So let's go back to the app and let's try and upload a file that isn't a Surrey object. So we have one kind of in this folder here. Now if we click run, we should firstly see our loading widget pop up, and then quite quickly we should see our modal message pop up. Let's have a look. Yeah, so error with file, there is an error with the file uploaded, see below for more details, invalid RDS file, which is correct. The file we uploaded was an R file, but it wasn't an RDS file. So we see this error and we can dismiss. And so this is really good for kind of front-end debugging. So you want to do a lot of kind of like try-catch blocks on the back end. Maybe not for this specific app, but if you're building apps, it's good to be able to debug on the front end as well, especially as the people often using these apps aren't going to be code savvy. So it's nice to have errors pop up so they know like what's gone wrong. Because if they try to upload a file and they were convinced it was an RDS file, but it wasn't, and there was no error message, they'd just be sat there stuck. So it's nice to tell them, look, this is why it went wrong. So that's our modal spinner. I should also mention that, so we have this modal pop up if there's an error. So look, we've got this if statement. If there's an, just picture this as being like if there's an error. If there's an error, we show the modal pop up. If there's not an error, we carry on. So I'm just going to put an else here, and we carry on. And actually what I'm also going to do just before this else block is I'm going to enable our button again, because remember we disabled it here, so we want to enable it again. So I'm just going to copy it and then change disable to enable. So in this else block we're basically going to carry on, assuming there's no errors. Now we're just going to carry on. So once we have loaded our Surrey object, we now want to produce some plots. But before we produce these plots, we want to create some new tabs to put these plots into. So as I mentioned, we want 2 new tabs to appear here, one for the metadata UMAP and one for the gene expression UMAP. So to create a new tab dynamically from the server block, we use a insert tab function. So insert tab, we need to add an ID, well, like an input ID. We need to add input ID, and this input ID is referring to like where these tabs are going to be put. So we want them next to our instructions tab. So we want our tabs to go in this tab set panel. It's the same tab panel where our instructions tab is. So what we do is we take the ID of this tab set panel, and in this case it's main tabs. We're going to take this ID and add it to here to our input ID. So we're going to be inserting this tab into the main tabs tab panel. Okay, and then we actually need to define the tab panel itself. So tab panel, we first define like a name, like what's the user going to see, what we're going to call this tab. I'm just going to call it UMAP for now, and then we can add a second tab panel, and this one is for our gene expression UMAP. So again, you want to put the same input ID, so we can just copy that from here, and don't forget to put a comma after that, by the way, guys. Input ID equals main tabs, comma. Sorry, I've absolutely butchered that, haven't I? So we're going to insert this tab, and don't forget to put a comma after the main tabs here. And for now, we'll also add our second tab in. So insert tab, and we can just copy this template. So this time we're going to call it, we'll call it gene expression. So if we pass the error check and we load in our Sirut object, we are going to insert 2 new tabs. Let's check that works. We refresh our app, we'll upload a Sirut object, and once that's uploaded, we'll click run. We'll see our widget pop up. And so on the back end right now, we have our Sirut object being loaded, but as you'll notice, the whole page has just gone dimmed, which means there's an error. So let's have a look at our terminal. Warning, error in insert tab, unused argument, input ID equals main underscore tabs. Okay, so let's have a look. Yeah, okay, my bad, guys. So it's not input ID with an underscore, it's literally input ID with camel case. So that was my error. Let's save that and try again. It can be quite annoying to like reload the Sirut object every time, because it does take like 10 seconds, but you gotta do what you gotta do. Maybe it might be useful to have like a test Sirut object that's very small, specifically for testing purposes, but I didn't think of that. Okay, so let's click on again. It says preparing plots, and on the back end it should be loading in our Sirut object. And yes, as we see, sort of behind the loading widget, we have 2 tabs that have appeared, UMAP and gene expression. However, this loading widget is still going on, and it will just go on forever because we haven't told it when to stop. So what we want to say is, once those tabs have been generated, then you can stop. So let's do that now. So insert tab, and then basically after this second insert tab, we are going to use a command called remove modal spinner. Remove modal spinner. And I guess we can also enable our run button here as well, because it will be disabled if it's past that test still. So let's try that. Refresh our app. Let's load the file and let's run. So hopefully this time we'll see this loading widget disappear, and yes, we do. So we have these new tabs and we can click on these new tabs. Nothing in them yet, but we now basically have the space where the new plots are going to be generated. So now let's figure out how we can actually display these plots in these tabs once they've been created. Okay, so for this, what we are going to do is, before our insert tabs sections here, we're going to use we're going to actually render our plots. So as I mentioned many times, the server function has this output parameter, and we've used the input parameter a lot. So basically input file, input reset, so our buttons, our run button, our reset button, our file, anything which how we're interacting with the app on the front end, that's our input. So our output is anything which is displayed on our app in response to our input. So the plots are going to be displayed on the app in response to our input, because we have to input what gene expression we want to see, what metadata column we want to see, and like what. So it's basically a response to our input, and so that's why the plot, instead of using, for example, input $file, the plot would be output $plot. So I'm going to demonstrate that now. So let's start with the UMAP that's going to show the metadata. So we can say output, and we can do output $plot, but let's make it more descriptive. We'll say output $UMAP, and we're going to use the render plot function. So this is built into our Shiny render plot, and here we're going to call the function which we created in global. So our function was called create metadata UMAP. We're basically going to put that function here. So we're going to call that function create metadata UMAP. It takes two arguments, our Sura object, so remember we defined that here, and then it's going to take a metadata column. Now we haven't actually set this yet, but I'm just going to put it in anyway as like a placeholder for now. So let's say metadata col, and this, it's not an input at the minute, but basically we're going to have a drop down in our new tab which is going to allow us to select a certain metadata column, and we should add that. In fact, let's stop there and let's add that to our tabs. So in each of these tabs we want to drop down, well, for the UMAP one we want to drop down for metadata. In the gene expression one we want to drop down for gene. Okay, so let me just make one of these for genes, so we have them both. So output $, let's just say, let's call this one the feature plot. We're going to do, instead of create metadata UMAP, we are running create feature plot. So this is our function, our global.r. We have our object, and then it takes a second argument which is our gene. So let's just do input $gene. That can be our drop down menu. Okay, that might be a little bit confusing. I'm sorry if I breezed over that a bit quickly. Bear with me, you're going to understand in a second. So input $gene and input $metadata col, they don't exist yet. We're going to create them in our insert tabs. Let me show you that now. So within these new tabs, the way I want it to be laid out is on one half we have our plot, and on the other half we have the drop down menu, which is going to basically say what metadata we want to display, or what gene expression we want to display over our UMAP. So I'm just going to code this out. Let's have a look. So firstly, for the UMAP, we have the tab panel UMAP. I'm going to put a comment comma here, and then we're going to start with a we can use a fluid row. So this is kind of HTML, I guess, where you can set like columns of certain width. We basically want 2 columns, one for the plot and one for the drop down menu. So we're going to say fluid row and then I guess our first column. And so if you're familiar with like HTML, you effectively have like 12 blocks along your page. So if you wanted your columns to be of equal width, you'd set each of them to be 6. I think we should set the plot column to be a bit bigger. So what I'm going to do is column width equals 8, and then beneath here we're going to actually plot our plot, but I'm going to leave that out just for now, just because we haven't actually made it yet. Well, we kind of have. It's output.featureplot and output.umap. In fact, yeah, we'll put it in there now. So width equals 8, and then we use a built-in function again of our Shiny called plot output, and we set an output ID which is going to match our plot. In our case, in this one, we've got output.umap, so our output ID is going to just be umap. So ID equals umap. And another thing we'll do while we're here is add a download button. So to be able to download these figures, we can use something called a download button. Again, built into our download button. And then you can set an ID for it. So we're going to set download umap, and then set like a name, like what's the user going to see. We'll just say, we can call it the same thing, download umap, but we'll add some spaces like that. And then we're just going to do exactly the same thing for this insert tab. So do we need that comma there? Yeah, so we want to add another column here actually, sorry. So we're going to add a column for our drop down menu. As I mentioned, the total width is 12, so if we've got 8 here, that means the width of this one needs to be 4. And we're going to have a drop down menu. So built into R, they have this thing called a select size input. This allows you to have a drop down menu which you can also search in. So because we've got so many genes in our SERP object, we don't want to just have to scroll through, because if we've got like over 1,000 genes, it's going to be so difficult to scroll through and find a single gene you want to have a look at. So we want the ability to search as well as actually scroll and find it. So we use something called a select size input. So select size input, and we're going to give it an ID. We'll call it metadata call, and then an actual name that the user is going to see. Metadata column. Let's put that on a different line. Okay, and then we actually have to say like what is going to be in the drop down menu. So for us in this one, it needs to be the column, the metadata column name. So because we've loaded in our SERP object as obj, we just do call names obj at meta.data. Okay, so that's one of our tabs, and then we're basically going to do exactly the same thing for our other tab, but obviously change it for different inputs. I'm just going to copy and paste it just because I'm a little bit lazy. So copy that. Main tab, so that stays the same. we need to call it something different. We're going to call this one, what was it, gene expression. It will be the same with 2 columns. The plot will be the biggest, but this time it's not UMAP. What is it? It's feature plot. So put feature plot there. Instead of download UMAP, we can just put download feature plot, and here just type it out for the user to see. Okay, then select size. We're going to call this gene and genes. The reason we're calling this gene is because InputJollyGene, that's like the name of our drop down. We're basically giving it that name in assigning it here, and so that's why here I use InputJollyGene. Okay. So that's quite a lot of coding without actually doing any testing. So let's save these files and let's go and refresh our app and see what the hell is going on. So let's reload in our Sirut object and then we'll rerun it. And we're getting an error because it's dimmed. Let's see what's the error. Error in plot output, unused argument ID equals UMAP. Okay, so plot output ID UMAP. Okay. Yeah, so sorry, it's not ID, it's output ID. So output ID with a capital I. So that should be the same for this one as well. And I think that should solve it. Let's just rerun it. So we get another error. What's going on this time? Could not find function select size input. Huh, have I spelled that right? Okay. Yeah, sorry, it's not select size input, it's select eyes. So we have to remove that S. So, my bad. Right, let's try a third time lucky. So if we run this, preparing plots, looks good to me. Okay, yeah, here we go. So now we have our drop downs, and okay, we've got an error there, but let's have a look. So we've got a drop down here with our metadata columns, great. We are seeing our UMAP, and let's say, okay, so we can't yet select different, oh, so it seems like we can select different columns, but not all the time. So end feature doesn't work, end count doesn't work, and %mt doesn't work. So if you remember in our global.r, these three columns in particular, we're creating a different type of UMAP. So that's telling me that there's an error, and I think the error is to do with the packages. We have to install some more packages. So what I'm going to do is I'm just going to paste in all the packages I used in this app, because I can't remember the exact ones which I used for those plots. Okay, so I'll just paste in all these here. Literally just go to my GitHub, it's in the description, and just copy these and just paste it into your global.r file, and I'm hoping now this is going to work. So I'm just going to quickly skip through the loading process. Okay, so now we have refreshed the app, run it again, and this time when we select these quantitative metadata columns, we can see they are being plotted as expected, and you can kind of see how they differ from the normal UMAP plots. So if we go on these normal plots, we just have, like, you know, binary values, colors, they're fine, but with these quantitative ones, this new way of plotting these UMAPs, we have this scale, so it's quite slightly nicer to look at, and so this seems to be working perfectly. And if we have a look at gene expression, okay, so what's going on here? Gene expression, this is not showing our genes, it's showing the metadata columns of the UMAP, so we obviously have an error there. And if we look in, yeah, so here in our insert tab, gene expression, because we just copied and pasted it, we've obviously missed something. Instead of col names obj at meta.data, it should just be row names obj. So the row names of our Sura object are our genes. So I'm just going to quickly refresh this. And this time if we go to the gene expression tab, yeah, our drop down menu now shows the genes, which is great. So if we can go through some of these, just pick random ones, it all seems to be working fine. There's a nice one that's being displayed very nicely. And let's try and click download feature plot, because I don't believe this is going to work yet. Okay, yeah, so that doesn't work properly. So this is the kind of next thing, and probably one of the last things we have to do. But apart from that, it's looking pretty good, I must say. That's a problem, because that seems to be crowding, so that's something to fix as well. But apart from that, let's fix these download buttons. So I know I already mentioned these kind of this download button function. So all this does is place the download button on the screen, but it doesn't actually tell, it doesn't tell RShiny what to do when we click the button. So we need to create a function, or two functions I guess, for each download button we have that tells RShiny what to do when we click download. So I'm just going to put those under here, and again we use the output, so we're going to put output dollar download button, so whatever we called it. So remember, so our output when we click the download button is it downloading to a certain location with a certain name on our file system. So what's the name of our output button? So for this one it's download UMAP, so we'll say output dollar download UMAP, and we use something called a download handler. So download handler, so we just assign that. And we say file name equals function, and we're gonna call, so this is basically we're setting the file name. So what is the file going to be called that we're downloading? And obviously we want it to change depending on what figure we're downloading. We want the gene name to be in the file name of our gene expression plots, we want the metadata column to be in the file name of our metadata plots. So we do that using the input value. So so for this one, whatever the metadata column is that's selected, so input dollar metadata call, that's going to be the first part of the file name, and then we're going to call it umap.png, I guess. So if our metadata column is cell type, it's going to be called cell type umap.png. And then the second part of this download handler, we need to create like what's the contents of the file. Like obviously we put the download button beneath the plot, but it doesn't actually link the plot to that download button. We actually need to link the plot and the download button, so that's what we're doing here with the content parameter. So we say content equals function which takes the file, so it takes a file. So our plot is going to be, we're going to create a plot again. So obviously global.r, we've had this create metadata umap create feature plot. We're going to call create metadata umap to be our plot, and this takes two arguments. One is the serial objects and another is the metadata column. So there's a plot, and then we finally have to save this plot, so we're just going to say ggsave file name equals file. We're just going to set some widths just to make sure it looks good. Width and height and type. That's our font. These are just going to make sure it looks decent. Okay, so that's our download button for the download umap. We basically do exactly the same thing for the download of the feature plot, so I'm just going to paste this below, but as you can see it's exactly the same logic. This time obviously we name our download button after the download button that we put on our page, and in a very similar way we name the file after the gene, so it's going to be gene feature plot.png. And then the content is going to be create feature plot of the object gene, and then we're saving it, so it's exactly the same thing. Okay, now let me reload the app and let's see if those buttons work. So I've run it. We'll start off by testing out the umap. Let's do the cell type umap. Let's click download. And there we have it. Our cell type umap, and as you can see in the top left here, the file is named after cell type, which is the metadata column. So that's great. And similarly, we should be able to do the same with this. What was that nice gene that had a, was it LXN? I cannot remember. Let's try LXN. It wasn't LXN. Whatever. Let's test the download button. LXN feature plot. Fantastic. And it kind of renders in this inverse color, like where the cells with high expression are in purple, which is kind of, I don't know, it's kind of like, I think it's like a default, it just does this, but I think it looks kind of cool. But yeah, anyway, I think we are almost there. Another thing, let's just quickly test the reset button works. In fact, before we do that, if you'll notice when I click these three dots, it this stuff kind of like pokes out. It doesn't look good, so we kind of want to fix that. which we'll do next, but let's just see if the reset button works. Okay, so the reset button doesn't clear the tabs, it only clears the input file. So there's two more things we need to do. One is fix this rendering, which doesn't look good, and two is we want the reset button to remove these tabs as well. So let's start with the remove tabs, because that's really simple. Basically, we already have this function. We already have this. So we're observing the event of the reset button. At the minute, when the reset button is clicked, all that's happening is we're removing the file input and we're disabling the run button. We basically want to add two more things to this. There's a simple function in R called remove tab. So we're going to do this twice. Remove the two tabs that we've made. So the first one, okay, the first argument here is the tab set panel. So if you remember, this is, where are we? This is this thing. So this is the tab set panel. We've got our instructions tab, and then we've dynamically created two extra tabs. We need to provide the ID of the tab set panel as the first parameter to the remove tab function. Where are we? Okay, remove tab. Yeah, the first argument is the ID of the tab set panel, and the second argument is the actual ID of the tab itself. Not the ID of the tab, sorry, it's the name of the tab that's displayed to the user. So one of them is UMAP. I think one of them is UMAP. Let's just double check. So inter tab UMAP, and the other one was gene expression. So let's just put that there like so. Okay, so that should handle the removing of the tabs when we press reset. The other thing that was going on is that when we minimize the sidebar, it was kind of overcrowding. It was kind of like the stuff in the sidebar was poking out. So to fix that, what we're going to do is we're going to add a few extra bits just in here. And I think what I'm going to do for simplicity is just remove the ability to minimize this tab, because I don't think we really need to have it to be minimized. We can just have it always like that, I think. So to do this, I'm going to basically add some custom CSS here. So this may be a little bit confusing. It shouldn't be too confusing. So I'm just going to type it out and then I can explain it. Okay, so here we basically added some custom, I guess, HTML CSS. So we're stopping the ability to toggle the sidebar. So I'm just going to reload this. I shouldn't have to actually load a file to see it working. Yeah, so basically there used to be 3 lines here where my cursor is, but now it's gone, so now we can't even minimize it. So that's removed the problem we were having. Okay, that is basically it. That is basically a very simple demonstration of how to build your own RShiny dashboard, which, for how simple it is, it's so useful. Like the speed in which you can just retrieve information from your Sura object is crazy. It's really useful app that probably took us, I'm not sure how long this video was, no more than 2 hours. So very useful thing to build, and as you can probably imagine, you can build out this app further. Like you can add extra tabs with different plots. Like there's heat maps, there's maybe like violin plots or whatever. There's so many different plots you can create with Seurat, and if you can build functions which can render those plots automatically, it's just a great way to explore your data quickly, and it can really like advance your kind of speed in which you do research. So yeah, as I mentioned, I'll mention it one more time, all the code for this tutorial is online. It's on my GitHub, which I'm going to link in the description. But one last tip I have for you guys is that if you are going to upload this code to GitHub, you want to make sure you add this Sura object to a gitignore file. This Sura object is very large, as we saw, like 300 megabytes, and so you probably don't want to be like uploading that to GitHub. So for those of you who don't know what a gitignore file is, it's basically a file you put in your directory. I'll just create it. It's like .git ignore, like that, and anything you put in here is going to be ignored by GitHub. So for example, in here, all you would do is type the name of this Sura object. So you just type like surat object.rds. So just do that. I'm just realizing actually it might be too late. Like, you might have already set up GitHub, but whatever, better late than never. So you just do that, and now that file won't be uploaded to GitHub basically. So that is it for the video. If you've stuck with me this long, then well done and thank you. I hope you've gained some valuable knowledge and experience from actually building out this project yourself. This was a fun one to build. You can really see how useful these apps can be. You know, although we've just built a very basic dashboard today, there's so much more scope to build it out further, add extra features, extra plots, and you can apply these apps to all sorts of different areas in bioinformatics, like data science, machine learning, or anything which involves data, which, you know, pretty much every aspect, every area in bioinformatics involves some sort of data analysis or data visualization. So building these apps to quickly do these analysis and look at results, very, very valuable. So look, I hope you've enjoyed the video. You know, put this project on your GitHub to display your skills. And yeah, if you've got any questions, drop a comment below, but preferably not like debugging questions. I don't want to be debugging too much. Just like Google it, ChatGPT it. But yeah, thank you anyway, and I'll see you in the next video.